So in today's video, I'm going to be going over AP Biology Unit 1 Macromolecules. Today's video, I'm going to be talking about the importance of carbon, how macromolecules form, and the four main macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Carbon is extremely important in biology and the formation of macromolecules because all of the macromolecules include carbon as a key component. Carbon is very special because it has 4 bonds that it can possibly form, which means that it can form a lot of bonds with a lot of different other atoms. So in many of the macromolecules, carbon will form bonds with hydrogen. It'll sometimes form bonds with oxygen. It just can do a lot of things, which makes it very, very unique and powerful. So the type of macromolecules that I'm going to be mentioning today, I already mentioned earlier. So macromolecules form mainly by dehydration synthesis. They are all made of monomers, so each of the monomers will go through dehydration synthesis and they'll come together to form what is called a polymer. Hydrolysis is the opposite of dehydration synthesis, which means that the macromolecules will be taken apart back into monomers. So I have images of how this process works here. So as you can see, dehydration synthesis is marked here and water is leaving. These are 2 monomers. Water is leaving, which is why it's called dehydration. Water leaves and then the bond is formed. This bond right here is hydrolysis, which is when water comes in. So it's the opposite reaction and the bond is broken. Carbohydrates is the first macromolecule I'm going to be talking about today. It's very important. It's used as energy for most of our cells. The monomer of carbohydrates is considered a monosaccharide. In other words, simple sugars. So most famous one that most people refer to is known as glucose. I have an image of a carbohydrate right here. Polymers are considered polysaccharides, which means it's just many monomers put together. So many glucoses put together would be considered a polysaccharide. And as I mentioned before, the sugars help store energy and it's easy access because it's easy for the body in the cell to break apart a polymer of carbohydrate and then they can get the monomer energy such as glucose easily. So these are types of carbohydrates that you need to know. So for storage, we have starch in plants and glycogen in animals. This is a polymer, which makes it easy for the animals to access. So when an animal or a plant needs energy, they will break down either a starch or glycogen into the glucose or the lactic acid, it depends, and then they will use that energy for the cell. And for structure, they have cellulose and chitin. So in plants, cellulose makes up the cell wall, which makes plants so rigid and it makes it help stand up, such as the stems of a flower or the big trunks of a tree. Chitin is in insects. Many insects will have them on their outer bodies, and it's also very solid. It's used as structure. So lipids are another type of macromolecule. Lipids are very unique. They're very weird also because they don't have one true monomer like all of the other macromolecules do, and they're really only grouped together because of one property. They are all nonpolar, which means they do not mix with water. And they're also not very big, so they're not really considered a true macromolecule, but nevertheless, they are considered one, and their function is also energy storage. But they store a little bit more energy, but it's harder to break down than carbohydrates. So when we think of lipids, we typically think of fats and steroids. Phospholipid is also very important, as I mentioned in my last video, and I'll mention phospholipids more in detail when I talk about the cell membrane, because that's really where it's going to come into play. So fats are made up of fatty acids and glycerol. These 2 things are often considered the monomers of lipids, and it's okay to think of this, but they're lipids that do not really have a true monomer, but they're typically made of fatty acids and glycerol. So fatty acids can either be saturated or unsaturated. Saturated means that it has a lot of the carbons around here, which means that it is going to be solid at room temperature. But unsaturated means that there may be 1 or more double bonds, which means that there's less hydrogen, which means that it's going to be liquid at room temperature. So this is a saturated fat and this is an unsaturated fat. Steroids we typically think of as cholesterol. Cholesterol is also important in the cell membrane, and I'll mention their function a little bit more later on. Proteins. So proteins are a very, very important macromolecule. I like to think of them as the most important macromolecule. Of course, we wouldn't be able to live without any of them, but proteins truly do a lot. The monomer is amino acids, and there's not that many amino acids. People are often surprised by that, but all the amino acids come together to make a polypeptide. And then the functions of protein, these are just a few. Proteins do a lot. Most famously, they do enzymes. They're also transport proteins, receptor proteins, and motor proteins, motor proteins play a role in cell division. They're the ones that will move the chromosomes to opposite sides of the cell. Receptor proteins are often on the outside of the cell, and they signal other cells to do other things, and they receive signals themselves as well. Transport proteins we'll talk about a lot when we talk about cell membranes, because that's usually where they're found. But they just move ions in and out. And the enzymes, of course, they help speed up reactions. So I'm going to talk about protein structure and how they denature. This is an amino acid. Amino acids are the monomer of a protein. They have an amino group, a carboxyl group, and an R group, as I shown in this picture right here. The green part is an amino group. This part, the yellow part, is a carboxyl group, and this is the R group. The R group is always changing. So everything about every amino acid is the same except for the R group. The R group can be anything, and that's what makes the amino acids unique. A peptide bond will connect the amino acids together, which is why the polymer is called a polypeptide. So there are 4 different structures of a protein, and most proteins will reach these 3 structures, and a select few will actually go to the 4th structure, but not all of them will reach this structure. Primary structure is simple. It's just a bunch of amino acids connected by peptide bonds. And the second structure can either be an alpha helix or a beta pleated sheet. And the structure is held together by hydrogen bonds. So the R groups will bind together, and then the hydrogens and oxygens of different amino acids will also bind together, and then they'll form one of these 2 shapes. Then the tertiary structure goes a little further because they'll have ionic, hydrophobic, and disulfide bonds. So that's just a lot of extra bonds that helps make them twisted a little bit more. And the quaternary structure is when multiple polypeptides are connected together. So people like to think of this as multiple proteins being connected together. So I have an image of what I mean here. This is a primary structure. As you can see, it's just a mini amino acids, these little purple balls connected together by peptide bonds right here. This is the alpha helix. Yes, this is the alpha helix, and this is the beta pleated sheet for the secondary structure. So hydrogen bonds are existing between these amino acids, and these amino acids to pull them together. In the B pleated sheet, it's the same thing. These hydrogen bonds and these hydrogen bonds. And then in the tertiary structure, there's a lot more bonds going on. So these bonds will bind together, and these bonds will bind together to where it forms into this type of structure. Over here, you can see that there's 4 proteins or 4 polypeptides bonded together to form a really large functioning protein. So the last macromolecule I'm going to talk about today is a nucleic acid. So the monomers of nucleic acids are nucleotides. And the most famous example of a nucleic acid is DNA and RNA. It codes for the proteins, and it's the basis of heredity, the basis of life, really. So this is a nucleotide structure. A nucleotide is made up of a nitrogenous base, sugar, and phosphate group. So as you can see here, this is what we call the phosphate group. There's typically just one, and then over here we have the sugar base. And then this can either be a ribose or deoxyribose, depending on if you're talking about DNA or RNA. And then we also have the nitrogenous base, which is expanded around here. So there's 5 different nitrogenous bases. So I don't know if many of you know this, but if it's in the DNA, we'll have the nitrogenous pyrimidine thymine. While if it's in an RNA, they'll have uracil. So typically, RNA has 4 nitrogenous bases, and DNA has 4 nitrogenous bases. These 2 are just switched out. So there are different kinds. A purine is an adenine and a guanine, while a pyrimidine is a cytosine and a thymine. So A purine will only bind to a pyrimidine, and the pyrimidine will only bind to a purine as well, so they cannot bind to each other. So adenine and guanine are not going to bind to each other. You learn more about this in the unit where we talk about DNA and heredity and all of that. This is just a quick touch-up. So DNA versus RNA, they're very similar in function, but they're also very different as well because they do different things and they go different places. So DNA typically only stays in the nucleus, while different types of RNA will leave and enter the nucleus as they please. So the sugar, as I mentioned before right here, the sugar base in DNA is a deoxyribose, while in RNA it's considered a ribose. So deoxyribose just means that the sugar has one less oxygen in DNA, while the ribose sugar will have more oxygen than the DNA. As I mentioned before, RNA has uracil and DNA has thymine. And then in DNA is considered double-stranded, as many of you might have seen, while RNA is typically considered single-stranded, in humans at least. There are some bacteria, some viruses that have double-stranded RNA, but in humans it's always single-stranded, and DNA it's always double-stranded in humans as well. And that's it for today's lesson. Please come back for my next video to learn more about that biology. I will be going over biology unit 1.3.